Hello and welcome back to the channel. Today we are going to discuss the topic Introduction to Genetic Study in Humans. The main objectives of today's topic are to know about the history of human genetics, to know about the basics of human genetics, to understand the meaning of heredity and variation, to know about the genes and chromosomes, and to understand the sex determination in humans. Let's begin with the Introduction to Human Genetics. As per the National Institute of Health, genetics is the study of genes and heredity. Several researchers have contributed to the field of genetics over time, aiding significantly to the understanding of the field, including Gregor Mendel, James Watson, William Bateson, Rosalind Franklin, and many others. The term genetics was first coined by William Bateson in 1905. Throughout this lecture, we are going to highlight some of the researchers and their respective contributions to the field of genetics. The major milestones in the field of genetics include the discovery of how traits get inherited, which is primarily credited to the research of Gregor Mendel. James Watson and Francis Crick elucidated the molecular basis of heredity through the discovery of DNA structure. And before that, Rosalind Franklin, an X-ray crystallographer and a British scientist, she is known for her significant contributions to the discovery of DNA molecular structure. In addition, the Human Genome Project focused on analyzing the human DNA in detail. Today, the field of genetics has shown tremendous advancements and has made the understanding of heredity and variation much easier. Let's now talk about Mendel and his contributions. Mendel cultivated pea plants, Pisum sativum, with various traits together. The laws governing the inheritance of heredity features from one generation to the next were developed as a result of his observations, and many of the ideas he expressed are still valid today. It was actually during the 19th century when Gregor Mendel performed the breakthrough research which was realized years after he passed away by some other workers. He grew pea plants in his garden to study the pattern of inheritance in them. His methodologies included the inbreeding of those plants that displayed varied traits, including height, and he bred the short plants with the tall plants to notice the inheritance pattern in pea plants. Mendel's research highlighted the inheritance of separate heredity factors and the passing of features from one generation to the next, or from parents to the children for the first time. Mendel introduced the ideas of genes being passed down from one generation to the next and the distribution of phenotypes and genotypes among the offsprings for the first time. Mendel also focused on 7 different morphological characteristics of garden peas. He examined multiple genes in the garden pea, each associated with a distinct trait, for example, the flower color or the texture of seeds or the height of the plant. Mendel postulated about the occurrence of certain heredity factors that were responsible for the traits he was studying. Today, we call those factors as genes. He further observed that the genes occur in 2 forms, and these alternate forms of the same gene are termed as alleles. He proposed that pea plants carry 2 copies of each gene, which may be similar or dissimilar. One of the copies is randomly incorporated into each gamete during the reproduction process. The female gamete unites with the male gamete at the time of fertilization to produce single cells, and those single cells are termed as zygotes, which then develop into new offsprings. During the gamete formation, the reduction in the gene copies occurs from 2 to 1, and it shows the restoration of 2 copies during the fertilization process. And it all occurs through the rules of inheritance which were discovered by Mendel. Mendel also pointed out that the genes are discrete entities. Through the process of hybridization, the individual alleles of a gene can be brought together in the same plant and then can be segregated from one another during the gamete formation. Mendel also discovered that the alleles of different genes are inherited independently of one another. These discoveries were published in 1866 but were noticed in 1900, 16 years after his death. His research was extended and applied to various kinds of organisms successfully, although with multiple exceptions. Today, his work has been used to study the inheritance patterns like the heredity studies in humans, chromosomal behavior, and others. Let us now learn about the significant contribution to the field of genetics by Watson and Crick. They revealed the chemical structure of DNA in 1953. In the middle of 20th century, genes were shown to consist of complex molecules called as nucleic acids, and these are further made of nucleotides. According to the hypothesis put forward by Watson and Crick, DNA molecules are made up of two chains of nucleotides that are joined by weak chemical bonds known as hydrogen bonds between different pairs of bases. Thymine and guanine bind with the adenine and cytosine respectively. These base pairing rules allow the sequence of one nucleotide chain in a double-stranded DNA molecule to be predicted from the sequence of the other. As a result, the two chains that make up a DNA molecule are complementary. Like DNA, RNA is made up of a chain of nucleotides connected to one another. Contrary to the DNA, RNA molecules are typically single-stranded in nature. The two strands of a DNA duplex are wrapped around one another in a helical form, or a helical pattern, as it was discovered by Watson and Crick. These molecules with helices can be enormous. Some are longer than 10 centimeters from end to end and contain hundreds of millions of nucleotide pairs. We could see them without a telescope if it were not for their incredibly thin nature. That means approximately 100 millionth of a centimeter. A nucleotide consists of three molecules: a sugar molecule, an acidic phosphate molecule, and a nitrogen-containing molecule. It's slightly basic in nature. The sugar that makes up the ribonucleic acid, or RNA, is ribose, whereas the sugar that makes up the deoxyribonucleic acid, or DNA, is deoxyribose. Within DNA or RNA, the nitrogen-containing base distinguishes one nucleotide from the another. The four different types of bases are adenine, guanine, cytosine, and uracil. Uracil occurs in RNA, and adenine, guanine, cytosine, and thymine occur in DNA. Thus, RNA contains uracil instead of thymine. When James Watson and Francis Crick explained how the nucleotides are organized within the DNA, it marked a major advancement in the field of nucleic acid research. Watson and Crick were previously aware that the nucleotides are connected to one another in a chain. The phosphate of one nucleotide and the sugar of another nucleotide interact chemically to generate the bonds, as you can see on the diagrams which are given on this slide. These interactions do not involve nitrogen-containing bases. A chain of nucleotides is made up of bases that are joined to a phosphate sugar backbone, one base for each sugar in the backbone. The bases form a linear sequence from one end of the chain to the other end, which is unique to that specific chain. This sequence of bases is what distinguishes one gene from the another. Thus, the structure of DNA, a macromolecule made up of two complementary chains of nucleotides, was clarified by Francis Crick and James Watson. In addition, many workers or researchers, including Pauling, Rosalind Franklin, Wilkins, Erwin Chargaff, have made significant contributions to the field of genetics. Let's now talk briefly about the contributions of Rosalind Franklin. She worked in the same laboratory as Wilkins. She studied X-ray diffraction to examine the wet fibers of DNA. She made significant advances in the X-ray diffraction technique with the DNA, and the diffraction pattern she obtained suggested several structural features of DNA, including its helical nature, more than one strand, as well as 10 base pairs per complete turn. So she was a pioneer to study about the chemical nature of DNA. Let us now have an overview of the Human Genome Project. The Human Genome Project, a world-famous research initiative that was started in 1990, was designed to establish the genetic, physical, and the sequence maps of the human genome by decoding the full nucleotide sequence. Identification of the genes dispersed across the genome was made feasible by the release of complete genomic sequence. The full genome sequence revelation, which has aided in the beginning of a new era in human genetics, had made it feasible to obtain information about genes, regulatory elements, and the chromosomal organization as well as structure. The Human Genome Project is a sequencing program backed by an international initiative to identify the order of roughly 3 billion nucleotide pairs in human DNA. The Human Genome Project was originally intended to entail international collaborations among researchers, with government funding for much of the effort. However, a privately sponsored initiative started by scientist and entrepreneur Craig Venter quickly came into existence alongside the government-supported project. All of these efforts culminated or ended up in the two extensive articles about the human genome that were published in 2001. According to those publications, 2.7 billion nucleotide pairs of human DNA were sequenced. The human genome, according to the computer analysis of this DNA, was estimated to include about or between 30 to 40,000 genes. The number of human genes has been revised to be around 20,500 in more recent studies. The location, structure, and possible functions of these genes have all been categorized. Studies are currently concentrating on how they affect various human qualities. Numerous other organisms' genomes have also been sequenced, including those of bacteria, fungi, plants, protists, and mammals. A large proportion or portion of this work was completed as a part of the Human Genome Project or other closely related initiatives. The sequencing efforts first concentrated on organisms that are particularly suitable for genetic studies. Now, what are the primary goals of the Human Genome Project? To sequence all the DNA in a human cell, that is, to establish the precise arrangement of nucleotides, adenine, thymine, guanine, cytosine, to identify whether DNA segments correspond to specific genes, that is, protein coding units. Thus, the Human Genome Project is an international initiative to unravel the human genome. Now, how was it completed? Thousands of volunteers provided their DNA samples, which were then delivered to the Human Genome Project sites throughout the world, where scientists performed DNA sequencing and analysis. Finally, a sequence draft was published in February 2001 in Science, Celera, a commercial corporation, and Nature, which through the public effort. After the complete sequence was released in April 2003, 50 years after Watson and Crick's publication of the DNA structure, scientists estimated that it included between 30 to 40,000 genes. We currently estimate that there are more like 25,000 genes. There is still much work to do in this field.